My seminar topic structural abbreviation in chromosome, deletion and duplication, presented by Neha. Structural abbreviation in chromosome. Structural abbreviation are the result of chromosome breakage and abnormal reunion of broken chromosome. They can be produced experimentally by exposing active cell to mutagen such as ionizing radiations. Four basic types: deletion, duplication, inversion and translocation. स्ट्रक्चरल एब्रीवेशन रिजल्ट होता है क्रोमोसोम ब्रेकेज का और एबनॉर्मल रीयूनियन ऑफ ब्रोकन क्रोमोसोम का. इसमें मिटोटिक रिकॉम्बिनेशन भी is processed by synapse of homologous chromosome. Mismatching can occur in this, particularly at chromosome site containing tandem repeats DNA sequence, etc. First, deletion and deficiency. Deletion occurs due to loss of a part of a chromosome. Small deletions are usually tolerated by an organism, but still can be seen as loop, forming bivalent at Purkinje stage of meiosis. It was first observed in salivary gland. Chromosome of Drosophila which remain in permanent state of pairing. Deficiency have an effect on inheritance also. Here is a picture showing loop forming bivalent at Purkinje stage of meiosis in Drosophila salivary gland. पिक्चर ऑफ डिलीशन. क्रोमोसोम में जीन है 1 ABCD, डी जीन डिलीट हो गया. उसके बाद में टाइप है डिलीशन के फर्स्ट टर्मिनल डिलीशन. ए सिंगल ब्रेक at the terminal portion of chromosome cause the loss of a genetic material and result in many physical abnormalities. Disorder. Wolff-Hishon syndrome. Wolff-Hishon syndrome, terminal deletion from short arm of chromosome number 4. Syndrome is characterized by intellectual impairment, microcephaly, development delay, abnormal external genitalia, large folded ear, and cleft of lips and palate. Degree of deletion and the variety of symptoms depend upon the amount of missing genetic material. Example of syndrome is caused by deletion of small arm of chromosome number 5. Cri-du-Chat syndrome occur due to the deletion of short arm of chromosome number 5. Anomalies including growth failure, abnormal face, severe mental retardation, distinctive high-pitched cat-like cry in infancy. 80% of cases chromosome carrying deletion comes from the father's sperm. Ring chromosomes are the distinctive product of terminal deletion from both ends of the same chromosome, which join together to form a ring chromosome in human. 2, 3, 4, 10, 14, 13, 15, 18, 20, 22 among other have been reported as a ring chromosome in humans. Ring chromosome result in characteristic abnormality including growth retardation. These ring chromosomes may be very small in size and may be present at extra chromosome in eukaryotes. ये तो टर्मिनल डिलीशन की फोटोग्राफ है. फर्स्ट पिक्चर में सेंट्रोमेयर से ऊपर जो टर्मिनल एरिया वो लॉस हो जाता है, उसको टर्मिनल डिलीशन बोलते हैं. और सेकंड पिक्चर में रिंग क्रोमोसोम की फॉर्मेशन दिखा रखी है, टर्मिनल से ब्रेक होके रिंग क्रोमोसोम की फॉर्मेशन. सेकंड पार्ट इंटरस्टिशियल डिलीशन, इसको इंटरकैलरी डिलीशन भी बोलते हैं. दिस एनोमली इन ह्यूमन वाज डिस्कवर्ड बाय बेंडिंग पैटर्न एनालिसिस ऑफ क्रोमोसोम एट प्रो-मेटाफेज स्टेज. They are caused by occurrence of two chromosomal break at the same time in terminal region, and this leads to the loss of small intestine chromosomal part. If there is a loss of internal piece due to break, it is called interstitial deletion. Intercalary deletion, loss of intercalary segment with two breaks. ये टू पार्ट के होमोजाइगस डिलीशन और हेटेरोजाइगस डिलीशन। होमोजाइगस डिलीशन सेगमेंट में इज लॉस्ट फ्रॉम द बोथ द क्रोमोसोम ऑफ होमोलॉगस पेयर, और हेटेरोजाइगस सेगमेंट इज लॉस्ट ओनली फ्रॉम वन ऑफ टू होमोलॉग। This deletion caused severe medical abnormalities, including growth defect, mental retardation, or even death of embryo. The embryo can survive if deletion of very small part of chromosome. A rare of interstitial deletion, XP21 of one chromosome in the long arm of X chromosome in males was reported to cause intellectual disability, short stature, hearing loss, etc. Disorder and cause rare interstitial deletion, ये भी बताया मैंने, reported to cause intellectual disability. Micro deletion are also type of interstitial deletion in which very small part is deleted. Such small deletion go medically undetected. Interstitial deletion from long of chromosome number 15 from the region just below the centromere causes. PWS is a congenital syndrome caused by deletion of part of long arm of parental chromosome 15 homologue and characterized by obesity, defective muscle tone, delayed growth, etc. PWS is a congenital syndrome caused by deletion of long arm of one parental क्रोमोसोम 15, और इसके करेक्टराइज हो सकते हैं ओबेसिटी, डिफेक्टिव मसल टोन, हाइपरजिया, हाइपरप्लास्टिया, जेनिटलिया, डिलाइड ग्रोथ, डेवलपमेंट, माइल्ड मेंटल रिटार्डेशन और इंटेलेक्चुअल एबनॉर्मलिटीज. एंड द अदर वन इज AS is congenital disorder from maternal chromosome which affect less than 1% in 20,000, causes movement and balance issue, delayed or absence of speech, mental retardation, combination of spontaneous outburst of laughter, smiling, and or a consistent happy demeanor, characteristic facial features, facial smaller than normal head, and mental retardation. इंटरस्टिशियल डिलीशन का शो कर रखा है, ब्रेक होता है इंटरस्टिशियल और इंटरकैलरी साइड से और लॉस्ट हो जाता है. Pseudodominance is also a part of deletion. Deficiency can make a recessive allele behave like a dominant allele. Pseudodominance. The principle of pseudodominance exhibited by deficiency heterozygous have been utilized for location of gene on specific chromosome in Drosophila, मेज और अदर ऑर्गेनिज्म. ये एक ऐसी डेफिशिएंसी होती है, इसमें जो रिसेसिव एलल होता है वो बिहेव करता है डोमिनेंट एलल की तरह. द प्रिंसिपल ऑफ सूडोडोमिनेंस एग्जिबिट करता है डेफिशिएंसी ऑफ heterozygote हैज़ बीन यूटिलाइज्ड फॉर लोकेशन ऑफ जीन सम स्पेसिफिक क्रोमोसोम. एग्जाम्पल्स में मेज दे रखा है. डुप्लीकेशन. Duplication are obtained due to the addition of a part of a chromosome. If duplication is present only one of two homologous chromosomes, then mitotic packaging cytological observation characteristic of deficiency will be obtained. Duplication occur in adjacent region at the displaced position of the same arm on a different arm of the same chromosome on a different chromosome, and sometimes the duplication may be found as a reverse repeat. Duplication on same are called tandem duplication. Duplication on different are called non-tandem duplication. Reverse repeat is also called reverse tandem duplication. And the tandem duplication that involves the end of chromosome is also called terminal tandem duplication. A prominent cause of duplication is X-ray, which cause deletion of one chromosome and corresponding duplication on its homologue. So reverse repeat is also called reverse tandem duplication. A prominent cause of duplication is X-ray, which cause deletion on one chromosome and corresponding duplication on its homologue. X-ray ब्रेक करती है क्रोमोसोम को, फिर एक्सरे वन होमोलोज पे, वन पीस पे, और नॉन टैंडम डुप्लीकेशन शो करता है। मतलब एक क्रोमोसोम से तो डिलीट होगा, दूसरे से डुप्लीकेट होगा। और इसमें शो कर रखा है ए पेयर ऑफ होमोलॉजिस क्रोमोसोम डुप्लीकेस हेट्रोजाइगस। इसमें 1 2 3 4 3 4 का डुप्लीकेशन है। पैकेटिंग कॉन्फ़िगरेशन, इसमें लूप बन जाता है। फॉर्मेशन ऑफ हेट्रोजाइगस, डुप्लीकेट हेट्रोजाइगस में लूप की फॉर्मेशन होती है। टैंडम डुप्लीकेशन शो कर रखा है यहाँ पर 1 2 3 2। टैंडम डुप्लीकेशन ऑन द सेम आर्म बट its place. नॉर्मल डुप्लीकेशन ऑफ डिफरेंट आर्म नॉन टैंडम डुप्लीकेशन ऑफ डिफरेंट क्रोमोसोम्स में, और ये रिवर्स रिपीट। लास्ट वन ई। Example, most dramatic example of duplication is Drosophila eye. Shape, bar eye is a character where eyes are narrower as compared to the normal eye shape. Bar character was due to duplication in region 16A of X chromosome. Reason of bar eye, formation of unequal crossing over due to improper alignment of chromosome. By the study of giant salivary gland chromosome, it could be demonstrated the bar character was due to duplication in region of 16A of X chromosome. Bar-eyed will be slightly different phenotyping in heterozygous or homozygous individual. Barred individual 16A give rise to the ultra bar 16A 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 3 times, and normal wild type 16A due to unequal crossing over. Even heterozygous and homozygous of bar eye can easily distinguish as the latter have smaller eyes. यहाँ पर देखो बार आई वाइल्ड टाइप का जीनोटाइप शो कर रखा है, उसमें बार टाइप को डुप्लीकेट हो गया, फिर डबल बार है 3 बार डुप्लीकेट होना। फिनोटाइप में चेंज हुआ है, बार आई जी डिफरेंट फिनोटाइप हो जाता है, फिजिकली आई में चेंज। The reason of bar eye formation is unequal crossing over due to improper alignment of chromosome, which leads to deletion in one chromosome and duplication in homologue. Similar event between two chromosomes having duplicate 16A leads to ultra bar eye formation. Wild type chromosome do not align properly and resulting in unequal crossing over. One chromosome has a bar duplication and the other a deletion. And then unequal crossing over between containing two copies of bar produce a chromosome with the bar copies, double bar mutation, and a wild type chromosome formation. Another example, Notch plus gene in Drosophila wing phenotype expression of duplication is abnormal. Normally this gene has two copies of single copy. Due to the deletion of other one leads to formation of notch in the wing of insect called tell positive, but 3 copies generate aberrant wing veins. देखो aberrant wing vein बनते हैं ये डुप्लीकेशन की वजह से नोच प्लस जीन में। Microduplication, just like microdeletion, they are hard to detect cytologically. Duplication of small portion of chromosome on short arm in chromosome 11 result in black with wide man, which is high birth weight, overgrowth of tongue and large kidney, hearing loss, and mental retardation. Thank you.